Hello, welcome to another Research Methods for the Biosciences screencast for R. This screencast covers section 10.6, Introduction to Parametric ANOVA, including section 10.7, One-Way Parametric ANOVA with equal numbers of replicates, section 10.8, Tukey's test, and section 10.9, One-Way Parametric ANOVA with unequal replicates. This test asks if three or more independent data samples can be considered to be from a common statistical population. A significant result indicates that this is unlikely and that there are significant differences between two or more of the samples. For example, Table 10.7 gives the fresh weight of the lobelia variety Hannah grown in one of 4 types of compost 8 weeks post-weaning from a tissue culture environment. The question is whether any of the compost promotes superior growth of the plants. There are 2 common parametric one-way ANOVA tests implemented by programs, and which one you will use depends on whether the variances are similar or not. We can test if the variances are similar, also termed homogeneous, by doing an F-test first on the data using the program. See Chapter 10 in the book and Box 10.7 for further details. One of the main reasons why your data may not have homogeneous variances is if you have unequal replicates. That is, unlike the data in Table 10.7, your samples have different number of data points. In general, when using these programs, do not worry about whether your replicates are equal or not. Focus instead on using the correct test for whether the variances are similar or different, as the program will use the correct statistical test for unequal sample sizes if required. If you get a significant result from the ANOVA, you may then wish to work out which samples are significantly different from each other. We cannot simply do a t-test for each sample pair combination, as this increases the likelihood of type 1 errors. Instead, there are many other tests collectively called post-hoc tests that can be carried out. In many cases, we prefer to use a test called the Tukey's test, although other tests, such as the Games Hull test, will be suggested in this screencast if required. This is the script we are going to be using, and in this case it is split over 2 screens. You may wish to freeze the screencast to look at it in more detail, or alternatively, you can download it from the Resource Center. The command functions are in black, are all in lowercase, and you must enter them exactly as shown. The lines in green are notes to help your understanding of how the script flows. The words in blue are variable names and can be changed to suit your data, but you must be consistent in spelling and the use of lower and uppercase letters. And the data are in red. There are several ways to load data into R. See my screencast Introduction to R for more details. Looking at the first variable, you can see that we have used the C operator to load the data into R. In the second variable, we have used the rep function, which will create a variable with each category or factor name in quotes repeated the number of times specified by each attribute before the program appends the next factor name to the variable. Here is the second page of the script. So let's run the script. First, track up to the beginning of the first line and click to place the cursor at the beginning of the script. You can now run the script line by line by pressing Ctrl R if you're using a Windows or Linux computer, or Command Option R if you're using a Mac. Each time we run a line, it appears in the console window, along with any results the command produces. So let's start to define the variables using the C operator. The first variable I define is plant weight. The plus sign in the console window indicates that I have split the data over more than one line, and I must run the next 2 lines to complete its declaration in R. For the compost variable, I am using the rep function with each attribute to declare it. If I now get R to display this variable in the console window by running just the name of the variable, you can see what this function does. To use the standard ANOVA function R, you must change the compost variable into a factor. We do this using the factor command. Now we have the data in R and we're ready to do the ANOVA test. But first, we need to decide whether our variances are homogeneous or not. We do this by running the Bartlett test. The Bartlett test assumes that your data is normally distributed. As we can see, the Bartlett test has given us a p-value of 0.2684. So what is the meaning of the p-value? A p-value of 1 means we can accept the null hypothesis as true, whereas a p-value of 0 means we can accept the null hypothesis as untrue. As we travel from a p-value of 1 to 0, the transition point between true and untrue is set at 0.05 in the biological sciences. The smaller the p-value below 0.05, the more confident we can be in rejecting the null hypothesis. A value of 0.2684 is above our transition value of 0.05, which means the result is not significant and we cannot reject the null hypothesis that states there is no difference between the variances of our samples. So in this case, we will use the results from the ANOVA test that assumes equal variances followed by the Tukey's post-hoc test. Some people recommend the use of the Levene's test to test the homogeneity of variances. The Levene's test can be found in the CAR package. For more information on how to load packages, please see my screencast on an introduction to R. So let's run the test. The standard ANOVA test in R is the AOV test. It produces a lot of results data. To simplify its interpretation, I'm going to save this information to the variable AOV.output and get R to give us the results we are interested in using the summary command. As we can see, the p-value is 0.00253. This is below our 0.05 transition probability, and we can say there is a significant difference between our composts and reject the null hypothesis. In fact, the difference is below 0.01, so we could conclude that there is a highly significant difference between the mean fresh weight of Lobelia variety Hannah when weaned on 4 different composts. But this does not tell us specifically which composts outperform each other. For this, we will have to look at the results from the Tukey's test. For this, we're going to use the command Tukey HSD and attach the AOV.output variable as the data source. The results are displayed in a table, with each row representing a comparison between 2 samples as detailed in column 1 and a p-value in column 5. For instance, row 1 compares compost B and A and gives us a p-value of about 0.003, suggesting that there is a significant difference in the growth of the tissue culture plants in these composts. So what if our sample variances were not homologous? In this case, we would use the one-way test, which is an implementation of the Welsh test. Even though it's not quite appropriate, let's run this test using the variables that we've already defined. Unlike the AOV test, the one-way test gives you the probability directly. In this case, it is 0.01932, a significant result, which would suggest that the composts are not all equal in supporting the growth of the lobelia plants. Obviously, we would now like to go on to run a post-hoc test to indicate which composts outperformed each other. The Tukey HSD test will not work with the one-way test output, so we need to find another method. One intrinsic method in R is to use the pairwise t-test. Now, as I said in the introduction, simply doing a t-test will actually increase type 1 errors. But in this case, we can adjust the p-value using the whole method to counter this. Here are the results. As you can see, it's given us a table summarizing the p-value for every comparison. So if we look at the top at cell 1, where it compares compost B to compost A, it gives us a p-value of 0.0035, a significant result. If you would rather run the game's HAL test, this can be found in the user-friendly science package. I hope you found this screencast helpful. For further information on how to use this test, or the theory behind it, then please consult the book. More information on how to use the program to perform the test can be found in our online web guides located in the Resource Centre. Thank you for listening.